GIS-α is a breakthrough dissolution apparatus developed to predict bioperformance of oral drug products. Today, in formulation development, the results of clinical and BE studies are often unpredictable by conventional dissolution apparatuses. This is largely due to the fact that conventional dissolution apparatuses are suitable for quality control and are not suitable to understand the bioperformance. GIS-α was developed to incorporate some features of the GI tracts into a dissolution apparatus to predict bioperformance of oral dosage forms. GIS-α was designed based on gastrointestinal simulator, GIS, which was introduced in papers at the University of Michigan, etc., with increased adaptability. GIS-α has the following features. Transfer model. GIS-α is not a conventional single-vessel apparatus, but a multi-compartment transfer apparatus, which allows the drug solution and suspension, moving from the gastric vessel to the small intestinal vessels, up to 3 vessels, to mimic the in vivo situation. Each vessel is associated with a specific section of the GI tract: the stomach, duodenum, jejunum, and ileum, and pH cab be monitored in each vessel. Biphasic model. GIS-α can also facilitate the setup of biphasic model to predict the bioperformance of certain drug products. GIS-α is effectively predict the bioperformance of drug product, which has pH-dependent solubility, BCS class 2 and 4. Approximately 40% of oral drug products fall into the low solubility category, BCS class 2 and 4. Moreover, up to 70% of drugs in the current drug discovery pipeline belong to BCS class 2, while another 20% belong to BCS class 4. This poses a significant challenge for researchers to understand the bioperformance and to optimize oral formulation for the assurance of the desired therapeutic outcomes. For this challenge, appropriate dissolution tests are crucial to predict bioperformance of oral dosage form in the GI tract and help researchers to optimize its formulation for the maximum bioavailability. Many researches have been published on the effective use of GIS-α in experiments, especially for class 2B drugs, where class 2B drug can be observed for its dissolution in the gastric vessel and its supersaturation, precipitation in the intestinal vessel. This observation allows the formulators to optimize the design of oral drug product. Moreover, the Product Quality Research Institute, PQRI Consortium, comprising over 10 research institutes, is conducting research to harmonize the biorelevant dissolution methodologies, and their findings highlighted the superiority of transfer dissolution apparatus in predicting bioperformance of oral dosage forms, which increasingly attract the interest of GIS-α. GIS-α equips 2 sets of GI systems and conducts 2 dissolution tests at the same time. 12 peristaltic pumps and paddles can be controlled individually. Thus, the transfer speed as well as paddle speed are adaptable and designable. The first-order rate of solution transfer rate can be automatically calculated based on the initial volume and designed gastric half-emptying time. In addition, pH probes can be attached to each vessel to monitor pH in real time. Sampling is possible with a generic Luer-type syringe through the access port from each vessel, as shown in the photo. Furthermore, the overall design is very user-friendly and easy. The peristaltic pump and pH probe can calibrated from the control box screen. The GIS-α has a capability of using up to 5 compartments, 5 different vessels, based on the researcher's individual experimental design. The large variety of vessel size and shape and paddle types are available, and the researchers can design the experiment the way they want. GIS-α, which is granted a U.S. patent, has been developed and manufactured by Canadivia Corporation to advance the field of biorelevant dissolution and the bioperformance prediction of oral dosage form. GIS-α is actively promoted internationally in the collaboration with innoreal-inc,